For many decades, scientists dedicated their lives and their careers to gathering DNA samples from all corners of the Earth. It was an unprecedented undertaking. Their collection included biological samples from indigenous tribes residing deep within the Amazon rainforest, nomadic groups traversing the harsh Mongolian steppes, isolated island communities scattered across the Pacific Ocean, and remote, high-altitude villages nestled within the Himalayas. These researchers meticulously sequenced millions of genetic markers. They were establishing the most comprehensive and complex database of human diversity ever assembled in history. They then conducted massive computational analyses designed to finally resolve a question that humankind had pondered for centuries. What exactly is race? Their discoveries dismantled every preconception we held regarding lineage, ancestry, and the very essence of humanity. The findings proved so radically astonishing that some researchers initially suspected their multi-million-dollar sequencing equipment had malfunctioned. The raw data revealed unexpected patterns and completely unforeseen genetic connections, producing revelations that dominated headlines in scientific journals globally. Let's delve into the actual insights provided by the raw data. The Human Genome Diversity Project launched in the early 1990s, driven by what was then a highly contentious objective. Its purpose was to gather genetic material from indigenous communities worldwide before the accelerating impacts of globalization potentially led to their disappearance. While some critics deemed it unethical, proponents asserted its critical importance for science. Yet all parties concurred on a single point: the resulting data would fundamentally alter our understanding of human variation forever. The project gathered samples from more than 1,000 individuals, spanning 52 diverse populations across 7 distinct geographical zones: Africa, Europe, the Middle East, Central Asia, East Asia, Oceania, and the Americas. The unprocessed data from this massive sequencing effort occupied vast server farms, amounting to petabytes of dense biological information ready for analysis. Upon receiving the initial computational results, researchers instantly observed a striking, undeniable fact. The genetic disparities among what society conventionally termed racial groups proved significantly smaller than anyone had anticipated. An individual walking the streets of Beijing and one born in Stockholm might exhibit vastly distinct physical appearances, including varying skin tones, bone structures, facial characteristics, and hair textures. However, at the microscopic DNA level, they shared an astounding 99.9% of their genetic blueprint. It was exclusively within the remaining 0.1% that the discoveries truly became fascinating. The subsequent analysis definitively demonstrated that our conventional, rigid racial classifications possessed virtually no genetic foundation whatsoever. For instance, the data revealed that an individual from East Africa could exhibit far greater genetic kinship with someone living in Southern Europe than with another person living just across the continent in West Africa. Furthermore, a Native American community isolated in the Amazon displayed entirely unforeseen genetic markers that are typically found only in populations originating from Papua New Guinea, thousands of miles across the Pacific. The clearly defined, rigid categories we had conceptualized for race were entirely absent from the genuine genetic data. They simply did not exist. Yet the most astonishing revelation of the entire genomic project was still unfolding in the laboratories. Researchers uncovered that the most extensive, profound genetic diversity on Earth resides entirely within the continent of Africa itself, rather than between continents or between what we historically designate as distinct races. Indeed, the genetic sequences prove that within the African continent, 2 individuals originating from adjacent Ethiopian villages might be more genetically divergent from one another than a randomly selected Ethiopian is from a randomly selected Norwegian. This observation, while initially counterintuitive, gains perfect evolutionary clarity once human migration patterns and our species timeline are fully understood. Homo sapiens emerged in Africa approximately 300,000 years ago. For the vast majority of our existence as a species, our ancient forebears remained exclusively on the African continent. During this immense expanse of time, diverse populations intermingled, dispersed, adjusted to varied local surroundings, and steadily amassed deep genetic variations over hundreds of millennia. Therefore, the genetic richness observed across the entire remainder of the globe represents, in essence, a mere subset, a tiny fraction, of the massive diversity already present in African populations, long before any significant exodus occurred. Approximately 70,000 years ago, when small, daring groups of early humans eventually journeyed out of Africa to explore the unknown, they took with them only a tiny portion of the overall genetic variation. In evolutionary biology, this phenomenon is known as the founder effect. A small pioneering population is fundamentally incapable of encapsulating the complete genetic diversity of its massive ancestral group. They constitute merely a localized, representative sample. This initial, limited sample then formed the genetic bedrock for all subsequent human populations in newly settled lands across Eurasia and beyond. Consider this analogy. Envision a bag holding 100 uniquely colored marbles, each symbolizing deep African genetic diversity. Now haphazardly select just 15 of these marbles and place them into a separate bag. This second bag epitomizes the genetic diversity that migrated from Africa. Even if these 15 marbles were to divide into various groups and disseminate across all of Europe and Asia, their total combined diversity would never exceed that contained within the initial bag of 100. The collected DNA data perfectly validated this trend along every single migratory path scientists charted. Human populations consistently exhibited reduced genetic diversity the greater their geographic distance from the African continent. European populations displayed less diversity compared to Middle Eastern populations. Similarly, East Asian groups exhibited less diversity than Central Asian groups. Native American populations, having undertaken the most extensive journey and crossed the freezing Bering Land Bridge during one of the final major migrations, demonstrated the lowest genetic diversity overall. However, overall diversity was not the sole unexpected finding hidden within the genomic data. Scientists unearthed an even more surprising revelation upon examining the specific genes that make us look different. The genes dictating visible characteristics such as skin color, hair texture, and facial traits constitute less than 0% of our entire genome. These are precisely the superficial traits we employ to classify race. These are the very characteristics that have fueled centuries of prejudice, systemic enslavement, and mass extermination. Yet they account for an incredibly minuscule, virtually insignificant portion of human genetic variation. Furthermore, these particular visual traits emerged quite recently in our human evolutionary history. The genetic mutation responsible for lighter skin tones in European populations surfaced merely about 8,000 years ago, a blink of an eye in evolutionary terms. Blue eyes developed even more recently, approximately 6,000 to 10,000 years ago, tracing back to a solitary, random ancestor living in the vicinity of the Black Sea. East Asian populations acquired their unique eyelid fold via a distinct genetic pathway, entirely unrelated to European features. These overt physical dissimilarities that society tends to emphasize are remarkably recent evolutionary adaptations to specific, harsh local surroundings. They are superficial modifications that transpired in a flash, relative to our species' complete historical timeline. If race lacks a foundational genetic basis, then what exactly accounts for the evident physical variations among populations globally? The explanation resides purely in localized environmental adaptation. As human populations expanded worldwide, they encountered vastly dissimilar climates, extreme elevations, varying solar radiation levels, and novel disease burdens. Natural selection promoted physical characteristics that aided immediate survival in those particular circumstances. Darker skin afforded vital protection against powerful, DNA-damaging ultraviolet radiation in equatorial regions. Lighter skin facilitated more efficient vitamin D synthesis in northern latitudes, where sunlight is scarce and weak. The epicanthic fold observed in East Asian populations might have offered essential protection for the eyes against harsh, freezing winds and intense glare from snow-covered landscapes. Amazingly, these adaptations arose independently in entirely disparate populations confronting comparable environmental challenges, a phenomenon termed convergent evolution. For example, populations in southern India and Aboriginal Australians both developed dark skin tones, despite their vast genetic distinctiveness and geographical separation. Their skin pigmentation evolved convergently because both groups inhabited areas with significant, punishing sun exposure. The specific genetic mutations responsible for dark skin in each population are entirely distinct. They ultimately achieved the exact same adaptive outcome by following varied, independent genetic routes. The genomic data also completely dismantled the long-held, dangerous notion of racial purity. A pure race has literally never existed in human history. There has also never been a truly isolated human population that refrained from intermixing with others, given sufficient time and opportunity. The fundamental concept of delineating strict genetic boundaries between racial groups crumbles under genomic examination. The majority of individuals possess genetic heritage originating from diverse, overlapping geographical areas. For instance, a person identifying strictly as Chinese might exhibit genetic markers linked heavily to Central Asian populations. An individual identifying as pure European could well carry deep Middle Eastern ancestral markers. Analyses of ancient DNA extracted from archaeological remains have shown that present-day Europeans are actually a chaotic genetic amalgamation. They possess genetic material derived from at least 3 separate ancient ancestral groups: early European agriculturalists, native hunter-gatherers, and Yamnaya pastoralists who violently migrated from the Eurasian steppes. None of these early groups were genetically pure either. They themselves were already complex composites of even earlier populations. This identical pattern of constant blending emerged in every single region scientists investigated around the globe. The genetic data unveiled another critical discovery that initially perplexed medical researchers. Certain genetic traits linked to deadly disease susceptibility indeed aggregate within particular geographical populations. Sickle cell anemia, for example, occurs much more often in individuals of West African descent. Tay-Sachs disease exhibits elevated rates in Ashkenazi Jewish communities, and cystic fibrosis impacts European populations disproportionately. While these patterns initially appeared to validate the old notion that race possessed medical and genetic significance, more in-depth genomic analysis presented the true explanation. These disease-related genetic variants did not disseminate due to racial distinctions. Their proliferation was strictly driven by the immense survival advantages they serendipitously conferred against particular localized threats. The sickle cell mutation, for instance, offers robust protection from malaria, a disease historically prevalent and deadly in West Africa. Possessing just a single copy of this gene granted life-saving malaria resistance without inducing full sickle cell disease. Consequently, natural selection heavily favored this mutation in areas where malaria represented a significant cause of death, irrespective of the arbitrary racial classification we might attribute to those communities today. This proves disease risks are geographic and environmental, completely severing the false link between race and biology. Therefore, if race completely lacks a biological and genetic foundation, from where did this pervasive concept originate? The rigid notion of distinct human races arose primarily during the 18th and 19th centuries. European scientists attempted to categorize human diversity using the exact same blunt methods they applied to plants and animals. They constructed artificial hierarchies founded upon superficial physical characteristics and cultural customs. They then baselessly ranked these races according to their perceived superiority. These fabricated classifications served a very specific geopolitical purpose: to legitimize colonialism, institutionalize slavery, and justify genocide. Although these early race theorists professed a strict adherence to objective scientific observation, their conclusions were entirely polluted by the biases, racism, and power dynamics prevalent in their era. They observed disparities in technological advancement and social structure, falsely attributing these variations to inherent biological inferiority instead of historical, geographical, and environmental factors. Contemporary genetics has utterly demolished these racial hierarchies, leaving them in the ash heap of history. Arguably, the most significant revelation from comparing global DNA is not what was discovered, but rather what was definitively not discovered. Scientists found zero genetic justification for national borders and absolutely no support for racial nationalism. Instead, the DNA analysis indicated an incredible truth. The average time frame since our most recent common ancestor is merely around 3,000 years. If you trace back just 120 generations, you would encounter an ancestor genetically linked to every single person living today. Your individual family tree and that of someone from the antipodal side of the globe converge at a singular point in the remarkably recent past. Recognizing that race is not genetically determined does not, however, nullify the undeniable, brutal social and economic repercussions of racism. The lived realities of individuals assigned to distinct racial groups diverge significantly today. The imbalances evident in wealth, health, and opportunity stem from historical and persistent social systems, not genetics. The scientists who analyzed our DNA provided us with the ultimate instrument to dismantle pseudo-scientific racism. They proved that underneath our superficial variations, we are one deeply connected human family, sharing a common ancestry and a unified genetic legacy. The illusion of race is shattered